After we've recorded it, we will edit and anonymize all of you guys, so you will be edited out. So you don't need to worry about the fact that you're going to feature in the recording. And we're just recording it for people who can't be here today, so that other people can watch the presentation. So Alan is here from the University of Cambridge. He's Director of Haematology. Is that right, Alan?
Consultant hematology.
Consultant, hematology consultant. And I guess 3 years ago, Alan came to the AAT with a proposal, along with his team, to investigate mutation timing in clonal trajectories in aplastic anemia. And today he's come back to explain not only what that means to everyone, but also what the team have done and what they've found out. Really excited to hear about this research. So I'm going to let Alan get on with it. But just to say that if you have any questions, hopefully we're all used to Zoom calls, and you can see there's a little button at the bottom called chat. If you have questions as we go along, you can write them in the chat. And then hopefully at the end, we're going to have time for a Q&A, and then I'll ask Alan your questions. So feel free to write those as we go along. And then at the end, I'll read out any questions in there and also hopefully take some from the floor as well. So yeah, without further ado, because you're not here to hear from me, I'm going to hand over to Alan.
Thanks very much indeed. So it's a great pleasure to speak to you today about, can you hear me all right? Is that the volume? Yeah. So yeah, it's a great pleasure to talk to you today about a project that you funded in our lab, and we're studying mutation timing and clonal trajectories in aplastic anemia. So what does that actually mean? So in terms of mutations, I guess if you think about the genetic information, that it consists of billions of letters of the alphabet, and so mutations are a change in one of those letters. It can be changed to a different letter, it could be deleted, or we could have letters that are added. And we're going to see, our aim is to try and understand the consequence of those mutations on the sort of life history of a blood stem cell in aplastic anemia. So that's what we mean by a clone. So it's a population of cells that are derived, from a single stem cell. So we're going to try and follow those in time and try and understand what the effect of the mutations is on, on the life history of these stem cells. No. So it's a problem changing this. Oh. So why did I get interested in aplastic anemia? So as a young doctor, I started life down at the Hammersmith Hospital in 1989, and I was introduced to all these luminaries of in the world of blood disorders. So that Ted Gordon Smith had actually gone off to St. George's a couple of years earlier, but he was still around at the Hammersmith. So people like Lucio Lutzato, John Goldman, and Inderjeet Dokal, who some of you may know already, who for his work on dyskeratosis congenita. So I met these people, and it was an incredible experience. I learned about the concept of bone marrow failure disorders, particularly the genetically inherited disorders, and also acquired aplastic anemia.
That's it.
So when we think about how aplastic anemia arises, the common sort of factor in aplastic anemia is fatty replacement of blood cells in the bone marrow, which you can see here. And this can arise in 3 different ways. One is physical damage, and the second one is immune destruction. So the immune system attacks the blood stem cells, and this is acquired. And then thirdly, we have constitutional genetic disorders, so disorders that patients inherit in the germline. And I'll talk about one such disease shortly. Shortman-Diamond syndrome. And there's a lot of overlap in aplastic anemia with other diseases, and this can present diagnostic challenges. So there's a lot of overlap between aplastic anemia and PNH, paroxysmal nocturnal hemoglobinuria. Sometimes it can be quite difficult to dissect aplastic anemia from myelodysplastic syndromes, or MDS, because they can present in similar ways with very low numbers of cells in the bone marrow. And as I said, a number of these so-called constitutional disorders can present with bone marrow failure and aplastic anemia. And I'll talk about one of those, which is a disease of the ribosomes. So ribosomes are machines in the cells that make all the proteins. And one particular disorder which we've been interested in for a while is called Shwachman-Diamond syndrome. And I'll talk about how we've used these sequencing technologies to learn more about which patients might go on to develop blood cancers in the context of SDS. And Indrajit Dokal studied these telomere disorders, which have shortened the ends of the chromosomes are short, and that can predispose to aplastic anemia in patients. A subset of them can go on to develop blood cancers, such as myelodysplastic syndrome and acute myeloid leukemia. So the question is, what are the changes in the bone marrow stem cells that might make some patients do worse, who might respond better? Can we design more targeted forms of treatment for patients with aplastic anemia so they can do better? And if we sort of think about the nature of aplastic anemia, so we were thinking about a concept called clonal hematopoiesis. So that just means that stem cells acquire a particular change in the alphabet and in one of the letters that gives them some sort of advantage that allows the cells to grow, to proliferate better, or escape the immune system, which is mediating all the damage to the bone marrow in aplastic anemia. So we can think of it in terms of this sort of bottleneck. So as we age, we all accumulate mutations. So some of the cells in the bone marrow are acquiring mutations over time. And then we have this insult, this immune attack on the bone marrow, which so we believe it's acquired. And then to escape from this bottleneck, the cells either acquire some sort of change in the alphabet that gives them a proliferative advantage, allows them to escape the immune system, or perhaps it's linked to what we hope is effective immunosuppressive therapy, which allows the bone marrow to then recover. So we'd expect to see different populations, different clones of cells as the marrow recovers, which might carry mutations that allow them to proliferate more rapidly, or which have mutations that allow them to escape the T cell immune attack. The T cells are a particular type of immune cell. Cytokines are the factors, substances that these immune cells can produce, which again can cause this bottlenecking, this reduction in the number of working cells in the bone marrow, and aplasia, so complete absence of cells in the bone marrow, which is a characteristic of aplastic anemia, which is the marrow is replaced with fat. And so in aplastic anemia, there are a number of common variants or changes in the alphabet. One is this PIG-A mutation, which is a characteristic feature of paroxysmal nocturnal hemoglobinuria. And then there are mutations in these factors shown here, ASXL1 B core. I can't actually see the, oops, have I stopped sharing? I've lost my screen, so can you still see the slides?
Yeah, we can still see clonal hematopoiesis in aplastic anemia.
Oh, here we are. Yep, good. Right. So yeah, so we have ASXL1 B core and DNMT3A. So that's three genes which affect the modifications of the alphabet. So these are mutations that can change the properties of the alphabet. They add modifications such as methylations, for example. So these are quite characteristic, and I'll show you examples of this later on. And then this thing called copy neutral loss of heterozygosity, which is commonly found on a piece of chromosome 6. So what do I mean by that? Well, it's loss of one copy of the DNA that is regulating the immune system. It's called this MHC locus. So PIGA and the 6P mutation are usually regarded as escape mechanisms that allow stem cells to escape the immune damage, whereas these mutations are associated with a proliferative advantage in the stem cells. And I'll show you patients where we see all combinations of these different mutations in the context of aplastic anemia. So, so what have you given us the money to do? So, as I said, we're looking at the timing of these alphabet changes and clonal trajectories. So we're looking at the life history of single stem cells in aplastic anemia. We want to try and understand how stem cell clones behave in different people with aplastic anemia. So hopefully we can design, what we call personalized approaches to treatment. So why do some people respond better to different treatments? Who's got a higher risk of going on to get complications like blood cancer? So this knowledge should hopefully enable us to design better treatments, identify complications earlier and give targeted therapies that are going to work best for the particular patient. We don't have a full picture of what the different so-called somatic or acquired mutations are in aplastic anemia, and this is a major barrier to developing these personalized approaches to treatment, or indeed, we lack understanding of what they do and how they might affect people's responses to treatment. So the solution is to try to apply innovative sequencing and bioinformatics techniques to try and learn more about the behavior of these individual stem cells before and after acquiring these mutations. So our aim was to try to map the mutations that are acquired in patients with aplastic anemia and PNH. And we want to reconstruct family trees of the blood system. And the main issue is, when are these mutations actually acquired in life, how the clones develop over time, and when and how the aplastic anemia develops. And this will give us a clearer picture of the dynamic behavior of stem cells following the acquisition of these mutations before and after treatment. And what I'll show you shortly is still work in progress. But yeah, we've made some good use of the money that you've provided to us. I hope you'll agree. So how do we perform these experiments? So we took blood or bone marrow from 10. We have initially examined 10 patients, 7 with aplastic anemia and 3 with PNH. We then expand these stem cells to get enough material to do what's called whole genome sequencing. So we basically sequence the entire alphabet of the genetic material in these cells. And by looking at the similarities and the differences between different clones, different stem cells, we're able to reconstruct these so-called family trees or phylogenetic trees, and time when the mutate. We can go right back in life to the very first cell division in principle. And this allows us to time when these mutations are actually acquired in life. And then we can do functional studies in a variety of model systems to try and interpret what we think is happening. And what sort of information can we get by mapping these somatic mutations? Well, by building these family trees, we're able to estimate the total number of changes in the genetic information. We can tease out signatures, which allows us to understand the disease processes that are happening in the stem cells. We can look at the overall level of clonality, and we can look at variants across and between different clones. And we can detect these so-called expansions of individual stem cell clones and identify these different mutations and try to interpret which ones are rescue mutations, which ones are good, and which ones are associated with perhaps poorer outcomes. And this is an illustration of a normal family tree from a 29-year-old. And you can see all these lines. So what we do is we can go back in time, because we all acquire mutations in our stem cells at the rate of about 15 a year, and it's a sort of linear plot. So the older we are, the more mutations we have. So we can go essentially estimate the age all the way back to conception based on the number of mutations that we have. And so a normal individual it's very, what we call polyclonal. It's very diverse. There's lots of different stem cells contributing to the blood system. So it looks very busy, lots of dark lines. And I'll show you the difference in the context of aplastemia and Shwachman-Diamond syndrome shortly. So, the example that I'm going to show you are data on this constitutional germline bone marrow failure disorder, which predisposes patients to developing aplastic anemia. So they can go on to develop blood cancers, and they have a variety of other clinical features which are characteristic of this disorder: pancreatic insufficiency and skeletal abnormalities. The problem is in Shwachman-Diamond syndrome, over 30%, we now think it's even up to 50% of these patients, unfortunately, go on to develop blood cancers. And at the moment, there are no disease modifying treatments. So if I just go back, so this is our normal family tree in a normal 29-year-old, but in the context of Shwachman-Diamond syndrome, this is an example of the family tree in one individual who was 22 years old when we initially sampled his bone marrow, and he's now, we had a second sample at the age of 24. So each of these dots is a single colony that we've performed whole genome sequencing. So we've sequenced the entire alphabet, the genetic code, if you like, in all of these stem cells. So we had about 140 stem cells in total where we've obtained this whole genome sequencing data, and this allows us to draw this family tree based on the similarities and the differences in the sequences that we can see. And we can map all the way from conception through to 22 years of age, what's happening with these clones of cells. And the most striking thing, as you can see, is it's very restricted in terms of the number of stem cells that are contributing to blood formation in this patient. So over half of the blood system is a contribution from only 2 or 3 stem cells. And you can see these what are called clonal expansions. So this is a stem cell that's come down here and then divided again and again. And so we have these large expansions in the bone marrow in a 22-year-old, and this is really uncommon. So you wouldn't normally see this under the age of about 70 years of age. And this reflects the very high degree of the selective pressure, because these patients have a defect in their ability to make proteins in the cell, because the ribosomes, the machines that make the proteins, are defective. So there's a very high selective pressure. So each of these stem cells, this line is a single clone. It adopts a different route out of the problem. So some of them acquire, so we've got these different mutations that are all mutually exclusive, but they're recurrent within a single patient. So we see 8 P53 mutations, 4 mutations in L5, and so on, L22 and EIF6, and we also see structural variants. We now understand that EIF6 is what we might call an adaptive mutation, so it allows the bone marrow to try to recover the ability to make ribosomes, whereas P53 is a bad prognostic indicator. And about 10% of the clones in this patient's bone marrow, they've acquired P53 mutations. We now know that acquiring just one mutation, P53, is not, it's possible that these clones persist for decades, but it's the acquisition of a second hit in P53 that causes the problem, as I'll show you in a second. So we can also, as I said, we can go back in time and estimate when these mutations actually occur in life. So in the case of SDS, we're able to show that many of these mutations are actually acquired even in utero, in the womb, and they occur all the way out to about 15 years of age in this individual. So they're occurring very, very early in life. And then the additional piece of information, we can look at the total number of mutations. As I said, each individual stem cell acquires about 15 mutations a year. So it's linear, the number of mutations is linear with age. And we see that in SDS, there's no, these patients still acquire the same number of mutations with time, but the exception was this individual here, where this clone has acquired 2 hits in P53, and this cell divides very rapidly over the course of about 6 months. And in this example, the patient presented with a blood cancer, in this case, myelodysplastic syndrome. And we're able to look at the so-called signature. So what are the properties of these mutations? And we could see that this thing, which is called SBS1, was more represented in this clone of cells. And it simply reflects the fact that these cells are turning over much more rapidly. So from the acquisition of this double hit to MDS was about 6 months. So it's a very limited window of opportunity here to do something about it, i.e. transplant the patient potentially. Okay, so that's SDS. That's what we can do with this technology, and I'm now going to show you what we've been doing in the context of aplastic anemia. But obviously this data is still work in progress, but I'll show you what data we have. So we've obtained, we're able to grow colonies from patients, but from 7 patients so far. We have another 7 aplastic anemia patients, and then 3 patients with PNH. And they range in, so we need to grow colonies to get enough material to do the whole genome sequencing. And in addition to that, for all of so this is 7, we've got colonies from 7 patients, but we also have granulocytes. So we've obtained DNA from the neutrophils, and we're performing what's called whole exome sequencing, this long read sequencing technique. So we'll be able to map the entire complement of acquired mutations in these patient bone marrows. And we're also going to look at the T cells as well. So we can broadly divide bone marrow cells into myeloid cells and lymphoid cells. And, we're going to learn a lot by, hopefully by combining sequencing data, both from the myeloid compartment, the granulocytes, and the T cells. So we successfully grew colonies even from patients with very severe aplastic anemia. So we have examples shown here. So this individual, we've got 26 colonies, and this individual 96 colonies, even in the context of very severe aplastic anemia. And so some examples of severe and non-severe as well. And I'll show you some examples of the phylogenies that we've obtained. So this is the first patient. This was a patient who had very severe aplastic anemia at diagnosis and was treated with standard, immunosuppressive therapy, anti-thymocyte globulin, cyclosporin, and this new drug, L-trombopag. I said before that mutations can be divided into 2 classes really. So commonly ASXL1 and these B-core mutations, which alter the modifications of the DNA, the methylation status. And we see in this individual, so that we're going here from birth through to 30, I guess he's 39 now. He's 35 at diagnosis, and these are individual colonies again that we've performed this whole genome sequencing. So in this individual, we have 3 B-core mutations and 1 ASXL1 mutation. And if we map onto this, the age of diagnosis, so what's quite striking is that we see that these mutations are, again, they're acquired very, very early in life, potentially even in utero, and they're persisting, even throughout life until the patient's diagnosed. We can already see that there's lots of changes in the bone marrow happening even before the patient's diagnosed. So, perhaps the immune process, I'd always tended to think that the immune process was happening within quite a limited window, maybe a year or two before the patient's diagnosed. But this kind of suggests that actually the disease process is decades long, potentially. And I'll show you other examples of this in a minute. So we've got relatively limited amounts of data after treatment here. The second patient was diagnosed at 37 years of age and 4 months. And so this was non-severe aplastic anemia, and he developed a PNH clone, which at the moment is around 22%, but it was so marked that he had to have anti-complement therapy in the form of humanized antibodies. But you can see, so we've only detected there's one large clone in this patient, which is causing all the clinical problems associated with PNH, hemolysis, increased risk of thrombosis. But what we see in the bone marrow by doing sequencing all these individual colonies is that this patient has 1, 2, 3, 4, 5 independent PIG-A mutations. This is the gene that gets mutated and causes loss of linked GPI linked proteins on the surface of the cell that makes them more sensitive to complement mediated lysis. So he has these PIG-A mutations, which we think of as an attempt to escape the immune system, because we think the target for the immune system is linked through one of these anchors, like a lipid phosphatidylinositol anchor on the surface of the stem cells. So we've got multiple stem cells trying to shed, whatever the target antigen is on the surface of the cell by mutating this PIG-A gene. And this individual also has DNMT3A as one of these mutations, which is also conferring what we think is a proliferative advantage. So by changing the methylation, the modifications of the alphabet in the genetic material. So this individual's got 2 types of mutations: these escape mutations and immune escape mutations, and these mutations that we presume are conferring a proliferative advantage. And if we then map when this individual was diagnosed, again, we can track the mutations all the way back to really early in life. And again, potentially these are happening even in utero, which to me was a real surprise, because diagnosed at the age of 37. So we see all these selection of these mutations and clonal expansions throughout life before he actually gets the diagnosis of aplastic anemia. And then this is another example of a patient with aplastic anemia who's been evolving to a blood cancer, to MDS. And what we see in this case, he was diagnosed at the age of 56. And this individual was 56 at diagnosis, and we can see that he has an ASXL1 mutation again, which is changing the modification of the DNA, and he also has this U2AF1, which is a splicing factor mutation, which is commonly, it's a feature of myelodysplastic syndrome. And again, these 2 mutations are acquired really early in life. This is conception all the way through to 56 years of age when he was diagnosed here. And again, we have all these clonal expansions that are happening, well before the initial diagnosis of aplastic anemia, suggesting that the disease process is happening, over decades and not restricted to a short window before the diagnosis. And then this is the last example. So this is a patient with aplastic anemia who was diagnosed at the age of 11. And she was refractory, so she had standard immunosuppressive treatment with anti-thymocyte globulin, cyclosporin, but didn't respond. And what you can see here is she has this 6p, which we talked about earlier on, 6p loss of heterozygosity. So that's we think is an immune escape variant. And she's also taken out another of the immune genes, HLA-B. So she's got 2 independent immune escape mutations, and you can see this HLA-B mutations associated with a huge clonal expansion. So the vast majority of the blood in this individual is contributed to by it's arising from this clone where the HLA-B locus has been disrupted. And again, the surprise, at least to me, is that is how early these mutations are arising in life. So again, this mutation's probably arisen within the first few months or even earlier in utero. And the process of clonal expansion has been happening throughout life until diagnosis. And then, there's a bit of, she's now responded well to danazol, so it'll be interesting to now go back and collect more data and see what's happened to these clonal expansions now that her marrow is essentially recovered. She's got normal blood counts. In fact, she's polycythemic. We had to reduce the danazol right down to 100 milligrams a week, and we may actually be able to stop it now. So yeah, so I think we've successfully, in this small cohort of patients, managed to grow colonies and perform the initial sequencing. There's still a lot more work to do, but it's opened up some really interesting observations, particularly about the timing of acquisition of these immune escape mutations. Perhaps in this individual, the fact that he had this U2AF1 mutation really early in life, it might suggest that there was a very strong chance that he might have evolved to MDS at some point. And maybe the diagnosis was MDS all along, although he did respond well to immunosuppressive therapy. So we need to go on and complete the sequencing and data analysis, and hopefully use this data as a pump priming mechanism to obtain a larger sum of money through a funding application to a larger charity or funding agency. I'd like to thank all of you for listening to me, and the Aplastic Anaemia Trust for providing this generous funding that's allowed us to do the work. This has been a collaboration with Jyoti Nangalia, Kudzai Naimondo, and Mike Spence Chapman at the Sanger, and Leeds Darren Newton and Richard Kelly. And I want to thank the clinicians and the nursing staff, my colleague Dora Ficanelli, Chi Wong, Duncan, Brian, and Phyllis Parson, who's our clinical nurse specialist. And thank you for your attention.
Thank you so much, Helen. Absolutely fascinating. I can see Ted clapping. Hopefully, everybody was following what Helen was saying there. It's absolutely fantastic. There's some big points for us at the Aplastic Anemia Trust. Firstly, this was funded by your fundraising and your marathon running and your donating. And it's a fantastic example of how when Alan's talking about pump priming larger applications, it wouldn't be possible to apply for big applications unless you have that first sort of information and spark that what you're looking at is useful and that you're finding out things from doing that work. And it's a really good example of how funding that early stage research can lead to potentially bigger and bigger things. I have some questions if no one else does, but I'll let anyone else ask them. Does everyone know how to raise your hand in Zoom? I have actually forgotten how to do that.
So I can speak for the Schwartman-Diamond syndrome, because that work is much further on. So what we've actually instituted is to monitor patients much more frequently. So we're doing full blood count monitoring every 3 to 4 months, because we have 2 examples where we could see these 2 hits in the P53. We could see the patient developing MDS, or in another case, an acute leukemia over a 6-month time frame, and we can quite accurately time the acquisition of that mutation to developing the disease, so we know that it's happening really quickly. So full blood count monitoring, and then at the moment we're doing serial sequencing, particularly looking for these P53 mutations, at least until we've got more information. We're doing this once every 6 months on a blood sample, because we know that there's a limited window for intervention once they've acquired those 2 hits. So that's informed our surveillance strategy. And the other thing was that we we got involved with the discussion with Genomics England on their newborn screening program, and so so many of these, maybe others in the audience have done this as well. But following the discussions, we've actually initiated testing in newborns. I mean, it comes with a bunch of ethical issues and potential concerns, but these are conditions, bone marrow failure conditions, where we could actually intervene early in life, specifically in the case of SDS with Creon, and just monitoring their diet, obviously within the first few months. So these are actionable conditions that we've now added to the newborn screening, and that's being rolled out. There's already about 10,000 patients where germline mutations have been identified, and it's now, we heard this week that it's going to be rolled out to all newborns over the next few years. So I think that's been quite a helpful practical step, newborn screening, as well as developing surveillance strategies, particularly for these high risk group of subset of patients. And that is going to come with the aplastic anemia. So once we have, I guess, identified mutations which we're particularly concerned about, we can institute better screening methods, more targeted surveillance, and better predictive algorithms, who's going to do well, who might not do so well, who would need to go to transplant potentially early on. Does that make sense?
Great.
Yeah, perfect. Thank you.
Tracy has her hand up. Hello, Tracy. Oh, you're on.
Hiya. Hi, Ellie. Hiya.
We've got you.
Yeah. So my daughter was diagnosed 6.5 years ago with aplastic anemia. She had ATG, cyclosporine, but could never be weaned off cyclosporine. Was sort of fairly well for 5 years, and then, well, she died last year after receiving 2 stem cell transplants, which failed. Now, you hear that it's not a genetic issue, but she has got a 4-year-old little boy. So would there be any merit in getting him checked or tested?
So you're sorry, I'm really sorry to hear about your daughter. That's really sad. So your daughter has a 4-year-old, is that what you're saying?
Yes, yeah. So in between sort of treatment, I don't know where it's a miracle. Don't know where he's come from with all the treatments she had, but yeah, he's now 4.5. And obviously you hear that this Fanconi anemia, you hear that sort of things, possibly genetic, but so I just don't know. Just listening to what you're saying there, would there be any merit in getting him checked? Whether that's a good thing or not, I don't know. It's just a question.
Yeah, I guess, yeah, I mean, if the child's well, then there's no need to look at the moment, but I mean, maybe further on, further down the line, it would be helpful to
Right.
investigate a bit further. Yeah.
Okay. Thanks.
We talked about this in the context of SDS screening in the clinic, and for mutations, and the pediatric doctors are not keen to do anything, certainly before the age of 7, in terms of screening. So yeah.
Okay. Thank you.
Ted.
Thank you.
Ted's waving.
Ted, would you like to ask a question?
Oh, yeah, sorry, I can't find the, various, I wasn't going to play a chime.
Don't worry, a physical one is fine.
Hello, it's very, really very interesting, extremely interesting, and of course, fantastic hard work looking at all those clones.
Yeah.
With Pig A, and which is slightly different from the other ones, like the P53 mutations and so on, in the way that it works as an escape mechanism.
Yeah.
You do find clones in very small numbers in a number of normal people going way back. You find Pig A clones in almost all aplastic anemia patients, but not necessarily expanded to clinical significance.
Yeah.
Do you think that there are a number of clones present at birth or in utero, because we all mutate quietly, which only become apparent when there is the drive to produce aplastic anemia maybe or probably immune function. Is that what happens? The clones are lurking. They're not the cause of aplasia from the beginning. They are. They become apparent because of the stimulus of aplastic anemia. Is that what you're suggesting?
Yeah, exactly. So we're all acquiring 15 mutations per genome per year, and so there's a heck of a lot of mutational burden, but those clones just don't get selected. So it's the environment, it's the immune attack which is allowing those things to be selected and expand and cause the problems. Yeah, absolutely. I think that's what the model. That would be the model. The thing that has really surprised me was how early they're getting selected. So I sort of naively thought that this process was over a few years prior to diagnosis, but it looks like that's not the case. That actually, this immune deregulation is present from really early in life. I don't know what you think about that.
That of course, is an interesting and perhaps new concept in relation to the pathogenesis of acquired aplastic anemia, leaving aside all the genetic types of dyskeratosis and so on and so forth.
It begs the question: is this an acquired?
Exactly, and of course,
exactly, and I think that's linked to Tracy's question. Is like those people here who've been diagnosed with aplastic anemia and told it's not a genetic condition, but then we're looking at these genes of people who've got aplastic anemia, and we're seeing these mutations happening in the womb. So, I understand, Ted, that you're saying that the mutations aren't causing the aplastic anemia, but then if that's happening so early, something
be
must
careful.
be.
There are a whole lot of well-defined genetic types of aplastic anemia.
Yes.
For example, a good one is dyskeratosis congenita, which may not present until adult life or later. And indeed, and I think, as Alan was suggesting, that if there is that gene there present as a cause of aplastic anemia, then the grandchild will show some hematological abnormalities long before the aplasia becomes apparent, and they should be investigated. But if the blood count is entirely normal and we follow it on, then there is probably no need to, and it has not been found in your unfortunate daughter, then there's no need to pursue that. But in acquired aplastic anemia, and of course, one of the things that we forget about aplastic anemia nowadays, because it's also exciting with the sort of work Alan is doing, is that there is often a trigger, which is a drug. Chloramphenicol was a good example. For example, no doubt that exposure to chloramphenicol in large doses increases the risk of aplastic anemia, maybe increases the risk of the immune response or whatever it is. Does that mean all aplastic anemias? No, they're not genetic. They really are acquired. I mean, it would be difficult to say and suggest that there is a hidden bank. There may be some hidden bank of genetic disorders which lead to aplastic anemia, which we haven't yet discovered, and that the acquired aplastic anemia triggered by whatever is really a reflection of genetic. I don't think we found that we can go as far as that yet, or at all, perhaps. A lot more to find out.
Sorry for what you've been through, but again, it speaks to the the deregulation of the immune system driving selection of these, mutations, variants, the changes in the alphabet and the genetic material that drives the problems. Potentially down the line, although just a small subset of patients may go on to get blood cancers, but it offers us the opportunity now to put more effective surveillance in place to intervene earlier.
What kind of a difference could that make, Alan, if we're talking about the small amount of people who have aplastic anemia who would go on to get blood cancer?
So I guess the definitive treatment would involve consideration of a bone marrow transplant early on if it's inevitable in that sense. I mean, with SDS, it's very clear cut. The biallelic P53 is going to progress to MDS cancer very quickly. So in that instance, it's clear. The discussion we should have early on is transplant a viable option? So I guess that's that's where we'd be headed. So, specific intervention to prevent that from happening until we can intervene if we know what the underlying mechanisms are, to intervene with the immune deregulation, or potentially correct the underlying molecular defect with a small molecule. And I guess transplants is the only thing we have at the moment.
Yeah. Something that's very familiar to me is people in our Facebook group, the AAT's Facebook group, asking about the decision whether to have ATG or a transplant, especially sort of adult patients, which treatment option they should go for. And this, I guess, this is the kind of information that could give individuals the idea about what treatment options might work better for them and might have more chance of success in the longer run.
Yeah. So it's part of the sort of diagnostic workup for sure.
Colin has his hand up.
Most centres that deal with these diseases have a multidisciplinary approach to diagnosing the condition. So we have input from, histopathology. We look at the morphology. We look at whether there are clones of cells carrying particular mutations, but particularly the morphology in the context of MDS. But if the bone marrow has a low number of cells, it can be quite technically challenging. But the treatment, I guess, initially might be the same: immune suppression. And many patients with so-called hypocellular MDS, I think Judith Marsh showed this originally, respond to immunosuppression. So there's no, in terms of a therapeutic intervention, there's no harm, at least initially, to try to get recovery in the counts. And maybe that's what's happened with our individual. So he responded really well to immunosuppression, but we still have the opportunity to, as time moves on, to better assess, what the actual underlying diagnosis was. But we have so many sophisticated tools these days, looking at mutation analysis, telomeres, etc. But the main thing is a multidisciplinary approach, so that we're pretty sure, and we'll decide what the best way forward is for each individual patient. I mean, usually in MDS, it's fairly obvious because the marrows tend to be, there are an increased number of cells. The morphology, the appearance of the cells looks very disordered. So yeah, usually it would be obvious if the counts started to fall again, and it's reassessment of the bone marrow further down the line. The disease is going to evolve over time. That's a good question. Yeah, I mean, I think over the next, certainly over the next 5 years, we'd expect that this would make an impact. We're obviously working with the guys at King's, and Austin has clinically annotated samples from these international trials. So it'd be really informative to go back and look at this in the context of a much larger cohort of patients. That's the crucial thing. These are quite small numbers, so we're keen to try and explore, increased numbers and better samples out, carefully annotated outcomes, where that would be potentially very informative. We need more resource, unfortunately, to use. There's quite a lot of work on this, and there's quite a lot of evidence that that's the case, that there is immune dysregulation in these particular populations of T cells. But I mean, the thing that we still don't know after all this time is what's the target for this immune dysregulation? What is the GPI linked to molecular molecules on the surface of the cell? So hopefully this sort of information will guide us closer to identifying the actual target for this pathological process. So yeah,
That's the dream.
Yeah. It's a good question about biobanking. I mean, hopefully you'll be close to a centre. If your own centre isn't biobanking, you'll be close to somewhere there is. But we're happy to see people. We have ethics approval that we have patients around the country who come and give samples, and we have cell lines. We've stored marrow material and DNA. So happy to consider if there isn't a biobanking facility available, we could happily have samples sent, or if you're happy to sign up to the consent, happy to obtain samples. I mean, we're talking all the time about setting this up, not just in the UK, but European wide. So biobanking repositories that people can share material and information, but it's a difficult process.
Yeah, and maybe we could add that to our website if you want to let me know the process of people maybe doing that if they have a biobank near them and they want to help. The other thing that's worth doing, Hardy, is just make sure, I mean, you're here, so maybe you are, but just make sure you're on the Aplastic Anemia Trust email list, because we do occasionally get asked to help people, recruit people to clinical trials. If they're looking for people with aplastic anemia or related bone marrow disorders, for that they will come to us and we will email you. So just it doesn't happen very often, but if it does, we will email you. So make sure that you're on the email list so you don't miss anything like that.
Thanks. I mean, letters of support are always really good. So the time when we put in the applications, we'd really welcome a letter from the Aplastic Anemia Trust supporting the work. I mean, that's really helpful. But yeah, we need to just knuckle down, and we're applying through. There's a CRUK one of these grand challenges, but I mean, obviously this is quite a sizable chunk of money, but we're going to throw our hat into the ring with the colleagues at the and see if we can get some money that way.
Thank you.
Need to be innovative and try to think big.
In fact, Ellen, I've got some lovely quotes from the patient panel about this piece of research, because I think you were on the panel when we considered this piece of research for funding, weren't you? So there was a panel of patients who filled in and gave their views, and I collected all those quotes. So we have some nice testimonials from patients about that already, and we could definitely email this group of people who came today and say, if you guys would like to say this is important and Cancer Research UK should fund it, we could definitely collect more feedback like that. We are out of time, so I am going to draw this to a close, but thank you so much for all of your thoughtful and brilliant discussions, and also just thank you to everybody who has ever fundraised or donated to the Aplastic Anemia Trust to help fund this kind of research, and who's doing that now, and who is part of our community and chose this research for us to fund and made it happen. It's absolutely fantastic to hear that what to the outside world might feel like a comparatively small investment in research can really turn up new information like this that can really make a difference. Thank you so much, Alan, for coming to sort of report back to our community on what's going on. We massively appreciate you taking the time.
Hopefully be able to update you again in the future once it's
great.
It's finished.
Please do let us know anytime you want to come back. We would love that. Thank you very much, everyone. I'm going to let you get on with your evenings. Thank you so much for being here.